Hello students, in our today's session we will be learning about the process of DNA packaging and also about heterochromatin and euchromatin. These are the concepts covered in the chapter Molecular Basis of Inheritance. So let's get started. Packaging of DNA. Length of DNA double helix molecule in a typical mammalian cell is approximately 2.2 meters. This can be worked out simply by multiplying the total number of base pairs with the distance between the consecutive base pairs. Approximate size of a typical nucleus is 10 to the power minus 6 meters. Then the question comes, student, how this long DNA molecule can then be accommodated in such a small nucleus. Have a look here. This is your chromosome, and then if it is further unwound, you get the supercoiled structures. These are the coiled. These are the nucleosomes over which your DNA is wound, and ultimately these proteins are called as histone proteins, and you have your DNA. So this DNA is a very long molecule, and we need to know that when you come and see this particular long DNA, it is tightly packed into our tiny small nucleus. So obviously for this structures to come, it must be condensed, coiled and supercoiled to fit into such a small nucleus. So now we need to understand how this process of packaging of DNA takes place. Students, if you remember, we have two types of cells, prokaryotic cells and eukaryotic cells. The packaging of the DNA in both these types of cells is quite different. So first we will see about the packaging in a prokaryotic cell. In prokaryotes like E. coli, cell size is almost 2 to 3 meters long. They do not have well-organized nucleus. It is without nuclear membrane and nucleolus. The nucleoid is small, circular, highly folded, naked ring of DNA, which is 1100 meter long in perimeter, containing about 4.6 million base pairs. The 1100 meter long, which is approximately 1.1 mm, if cut and stretched out. The nucleoid is to be fitted or packed into a cell which is hardly 2 to 3 meters long. Hence, the negatively charged DNA becomes circular, reduced in size to around 350 millimeters in diameter. Here, have a look at this figure. The DNA takes a circular shape. The DNA takes a circular shape. This further reduces to 30 micrometers in diameters because of foldings or loops. Here, these are the foldings and the loops that get formed. 40 to 50 domains or loops are formed. These loops should come out to be around 40 to 50 in number. Formation of loops is assisted by RNA connectors. So, you require an RNA connector for the formation of these loops. Each domain is further coiled and supercoiled, thereby reducing the size down to 2 microns in diameter. The next step. So, it's supercoiled and it's folded into the chromosomes. This coiling, that is packaging, is assisted by positively charged histone-like DNA binding proteins. Proteins and enzymes like DNA gyrase and DNA topoisomerase 1 are required for maintaining these supercoiled state. So this is how the packaging of DNA takes place in a prokaryotic cell. Students, now let's have a look at how the DNA is packed in eukaryotes. Eukaryotes show well-organized nucleus containing nuclear membrane, nucleolus, and thread-like material in the form of chromosomes. In the chromosomes, DNA is associated with histone and non-histone proteins, as was reported by R. Kornberg in 1974. The organization of DNA is much more complex in eukaryotes. Depending upon the abundance of amino acid residues with charged side chains, a protein acquires its charge. Histones are the proteins that are rich in lysine and arginine residues. Both these amino acid residues are basic amino acids and carry positively charge or a positive charge with them. So histones are a set of positively charged basic proteins. These histones organize themselves to make a unit of 8 molecules known as an histone octamer. Have a look at the diagram here. You can see these round structures. These are your histone proteins. You have 8 such proteins which come together and they get arranged into a structure which is called as a histone octamer. The negatively charged helical DNA is wrapped around the positively charged histone octamer. So you have the DNA here. It's negatively charged. It gets wrapped around the histones that are positively charged, and once this winding takes place, the structure then formed is known as a nucleosome. The nucleosome core is made up of 2 molecules of each of the 4 types of histone proteins. So you have 2 each, that is H2A, then you have H2B, and you have H3 and H4. You have one H1 protein that binds the thread of the DNA from where it enters and from where it leaves the nucleosome. Even have a look at this picture here. You can see how the DNA gets wrapped out to the octamer in 2 turns and then comes out and goes to the next octamer, and the DNA which is present between the 2 nucleosomes is called as the linker DNA. Students, moving further with the process of packaging of DNA in eukaryotes, once the nucleosome is formed, one nucleosome approximately contains 200 base pair long DNA helix wound around it. About 146 base pair long segment of DNA remains present in each nucleosome. Nucleosomes are the repeating units of chromatin, which are thread-like stained colored bodies present in the nucleus. They look like beads on strings when observed under the electron microscope. This picture here shows us an image of the nucleosomes in an electron microscope. You can see it appears like beads are arranged on a string. The black arrow points towards one nucleosome or an histone core, and the white arrow points out to the linker region which connects two nucleosomes. DNA helix of 200 base pairs wraps around the histone octamer by 1/3 turns. 6 such nucleosomes get coiled and they form solenoid-like looking structures which appear like the coiled telephone wires. The chromatin is packed to form a solenoid structure of 30 nanometers diameters, which is also known as 300 angstroms, and further supercoiling tends to form a looped structure called the chromatin fibers, which further coils and condenses at metaphase stage to form the chromosomes. First, let's have a look at this figure here. That is where the wrapping of your DNA is taking place. When your DNA takes this to turn, around 146 base pair of DNA gets wrapped up and the remaining are entering and leaving the DNA. In the linker region also you have certain DNA that is present. Students, the packaging of chromatin at higher levels need additional set of proteins that are called as non-histone chromosomal proteins. These are additional sets of protein that contribute to the packaging of the chromatin at higher levels. So a quick recap, this is your DNA which gets wound to your octamers and then it further gets coiled to form your solenoid structure which looks like the telephone wires, and then you have the further coiling that takes place till it forms the chromatin fibers. Here also you have a different way of explaining it out. Your DNA is there, then it gets arranged to form the beads on string structure, then it gets further coiled and extensions are there, where every time the length of the DNA is getting reduced till it reaches to a size of the most condensed form, that is 1400 nanometers at the time of the metaphase stage during your cell divisions, where your chromosomes are most condensed and are ready to undergo the stages of your mitosis. Students, let us see now what's a heterochromatin and a euchromatin. Heterochromatin. In eukaryotic cells, some segments of the chromonema or the chromosomes during interphase and early prophase remain in condensed state. These regions constitute the heterochromatin. Here, if you see this dark stained regions, this is called as the heterochromatin. This term was proposed by Heaps. These regions are localized near the centromere, the telomeres, and also intercalated. This is the centromere region and this is the telomere region. It is genetically most inactive. It stains strongly and appears dark. Heterochromatin, however, is 2 to 3 times more rich in DNA than the euchromatin, but it is inactive. So it stains dark because it is quite condensed. And the other region is the heterochromatin region. After heterochromatin, we have the euchromatin region. The regions of chromonema which are in the non-condensed state constitute the euchromatin. Euchromatin region stains light. Euchromatin is genetically very much active and fast replicating. Have a look at this figure here. You will see a region here which is stained lightly. This region is called as euchromatin. It is less condensed and it is rich in genes, especially it has higher GC content, and since they are little loose, they stain lighter, and they are the ones who are very much active and involved in replications, faster replications. Euchromatin is transcriptionally active, while heterochromatin is transcriptionally almost inactive. So, transcriptionally active region is the heterochromatin. This information will be particularly important when you will be studying the process of transcription further in this chapter. So, there are two regions in your chromosomes, heterochromatin and euchromatin. The heterochromatin region again is quite condensed and it is genetically inactive, and the euchromatin region is less condensed, it stains lighter and is genetically more active. So students, just a quick recap on the two types of packagings that we have seen. That is, first is the packaging of DNA in prokaryotes. Simple, you start with a circular DNA. It then gets folded into the supercoiled structures and forms these loops. These loops further get supercoiled and have an RNA to connect it, and the further supercoiled structure is present loosely suspended in the nucleus, as against the ones that we will see that we just saw in our eukaryotic packaging. In our eukaryotic packaging, we had a DNA which gets wound around the histone octamer, forming a beads on ring structure. It further forms your solenoid fibers, which further gets even more supercoiled, finally to give you your two chromatins, which you see under the microscope, which are visible under the microscope at the time of metaphase. Thank you students for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe and keep learning.